I guess it just feels free to welcome you all to the 25th IIT seminar, where Bob Holt of the University of Florida and author of innumerable papers and books will be presenting. Please do keep your microphones muted until questions at the end, but feel free to keep your cameras on if you want to give a bit of an audience. And so Bob, over to you.
Yes, well, first I'd like to thank Chris and Giza and Axel and any other organizers for inviting me. This has been a great series. And this wasn't scheduled, but I'd like to put in a free advertisement for a really good book by Giza and Liz, since Liz is on the call, Theory-Based Ecology, Oxford University Press. I think all theoretical ecologists ought to get a copy and read it and have their students and postdocs read it. That's what I do in my lab. So having said that, what I'm going to do now is turn to my talk. And I titled it COVID-19 Meets the Hutchinsonian Niche, which proves I am not a public health person, because talking to one of our epidemiologists, who I'll mention later, he gently pointed out to me COVID-19 means the disease and the actual organism is SARS-CoV-2. And I've given versions of this talk, Columbia University, Nick Cortezes, who's honorary postdoc with me now, gave a talk related to this in Case Western, and then I was out at Utah and gave a talk a few weeks ago. And so I'm giving you a version of that. And the way this started was, and I don't know why, but it's not allowing me to.
Bob, you don't share your screen at the moment, just so you know.
Oh, I don't share the screen?
Yep.
Oh my goodness. Okay, sorry about that. I knew there was something wrong. Okay, so let's try. Yeah, I skipped a step. Must have gone out of this and then back in again. Okay, can you see that now?
Yes.
Yes, okay, great. So as I was saying, this is a version of talk I've given recently, and hopefully okay. And just to say who I am, I'm an intellectual generalist. I work on lots of problems. I'm a theoretician. I'm not really a mathematician. This will not be a heavily mathematical talk. Theory includes models, but it's more than models. I worked on, for those of you who don't know me, a lot of things involving biodiversity dynamics, apparent competition, intragill predation, and so forth. I've done a lot on spatial ecology. I've done a big field experiment in Kansas having to do with the interplay of habitat fragmentation and secondary succession. I've done a lot on the theory of range margins and infectious disease ecology, and from an evolutionary point of view, niche conservatism, rapid evolution, including at range margins. And this talk involves infectious disease ecology, but also spatial ecology and ideas about the niche. I'm going to be trying to relate this. And the way this material came about, the recent material, has to do with this group of people right here who are associated with my lab. Nick, who's sitting behind me, postdoc, Maggie Simon, another great postdoc of mine, my long-term research associate, Mike Barfield, he's been with me for 25 years. We've written 50 plus papers together. And two people in our Emerging Pathogen Institute, Bert Singer, who's National Academy member, very esteemed epidemiologist, and Greg Glass, who's also in epidemiology, but he started out in small mammal ecology. He was my first graduate student back in Kansas in about 1980, long time ago. So we go back a little over a year to the start of the pandemic, and some things were happening. My epidemiologist colleagues were telling me in January that the world had to really worry about things that were looming on the horizon. And of course, early in the pandemic, this shows some data from the BBC showing on a log scale on the y-axis against time, linear increases in the number of cases, which is of course a hallmark of exponential growth of invasive species. And our university shut down in mid-March. And then Maggie, who I just showed you a picture of, so we weren't allowed to come to campus. She sent out an email and talked to me and says, Bob, we're a bunch of theoreticians. Can't we do anything that's relevant? You know, we're working on all kinds of problems that have nothing to do with this pandemic, but surely we can put our minds together. And then Greg Glass approached me a few days later and he says, hey, Bob, your early work on persistence of a metapopulation consisted entirely of sinks. that vary over time could be very relevant. And so what I'm going to be doing is giving you a story, walking back through some historical things, relating it to niche concepts, and so forth. So here's some papers. I'm going to quickly go through this this material because this background. This was all purely theoretical exercises, but we think it's very practically relevant to what's actually happened in degrading pandemic control around the world. And the fruit of our work last year appeared in PNAS in December, and we've got some follow-up work that we're chewing on now and trying to write grant proposals and so forth. And so I'll be talking about the pandemic, but I'll be going back to this earlier work, kind of metapopulations, and splicing it with some ideas that have to do with just basic niche concepts. So people who aren't really that interested in infectious disease ecology still might find some value in what I'm saying. And I've got an essay forthcoming in the American Naturalist revisiting ideas I put out in 2009 in PNAS on refining niche concepts. So I'm going to talk about niche concepts for a few minutes, and then we're going to go back to these other issues of metapopulations, and then finally the pandemic. So what do we mean by the niche? It's a complicated term. You know, some people think it's so complicated it ought to be dropped entirely from the ecological lexicon. I think that's too strong. But if you go back to its origin in the English language, it meant a place in the environment like this this niche in the church holding a statue, literally a physical place that when occupied by one statue may prevent other statues from crowding into the same place. And Hutchinson, in his famous 1958 concluding remarks, abstracted this by saying the niche describes states of the environment that permit a species to persist indefinitely. And I'm going to come back to this statement right at the end of the talk. And of course, his original concept was set theoretic, right? And he was interested in community ecology. And I'm not going to be talking about community ecology, impacts and feedbacks and so forth very much. I'll be talking about another simpler meaning. So he was interested in do niches overlap? Is that what they did, competitive exclusion and the like? Well, his set theoretic idea still lives on in the cottage industry called ecological niche modeling, where you take distributional data for species like this fly-catching bird, the eastern phoebe, and you look at its winter range, and you can see if you plot the average minimum January isotherm on it, it matches pretty well all the way from New Jersey to El Paso, Texas. So there you have a first order so-called ecological niche model that can describe where you expect to find phoebes in the winter and where you expect not to find phoebes, okay? And that's sometimes that's all you can do. And I'll touch on that a little bit relative to COVID in a second. So then 20 years later, Hutchinson refined, you know, he altered his niche concept by relating it to the most basic concept in population biology, which is intrinsic growth rate, exponential growth, little r, per capita births minus deaths, particularly at low densities. And he was relying upon this man, Bassett McGuire, who most people have never heard of, professor at Austin, Texas, had some very interesting papers on these ideas in the 70s. So here on the right, we have per capita instantaneous birth rates as a function of two abstract environmental variables. It's the mountain bulging out of the screen at you, then the death rate, same two variables, and it's a hole going into there. And the difference between them is a niche response surface, okay? And where little r is 0, that's what we call the niche. So basically, the Hutchinsonian niche, as in Hutchinson 1978, is some measure of demographic performance or absolute fitness, like intrinsic growth rate, of individuals or aggregates of individuals, because you could do this for population of species or even entire clades, expressed as a function of the environment. And of course, if you're talking about infectious diseases in humans, the environment includes behavior, socioeconomic factors, and so forth. We'll talk about that a little later on. And so it's abstractly a mapping of population dynamics for a given phenotype or lineage onto an abstract environmental space. It's a whole surface, but the edge of this surface is, of course, often of particular interest. And but what you mean is a habitat with conditions A, that temperature and humidity, you introduce a reasonable individuals you expect them to persist, you put them outside of it, and it goes extinct. Very simple, okay? Now, here's my favorite example of the empirical quantification of niche. And actually, this is mentioned in Giza and Liz's really fine book that I mentioned at the start of the talk. This is my friend Richard Sibley, who's down in Reading, England, kind of outside of London, and he was working on actually he had some works involving ecotoxicology where this was relevant. He's looking at this zooplankton, Daphnia magna, which lives in water bodies. If you go up to Yorkshire, this is all the water bodies of any size in Yorkshire, ponds and lakes, okay? But if you look at where Daphnia magna occurs, it's a much smaller subset, but it's scattered over all of Yorkshire, so it's not likely dispersal limitation because of glaciation or whatever. So his hypothesis was in some way distribution reflects the niche. And so they brought a clone of Daphnia magna into the lab and exposed them to water with ample, ad libitum food. Water chemistries that varied in pH, and osmotic balance is really important for any aquatic organism, and calcium concentration, which apparently influences their tolerance of pH. And so they came up with a niche response surface, a very nice one. Okay, and then the bottom graph shows the dashed line. On the left, little r is negative in the lab, and on the right is positive. And there was great predictive value to this lab measurement of two niche components, because almost always if little r was predicted to be negative, you didn't find Daphnia magna. There was one counter example, and it was a pond right next to another water body with a really different water chemistry. Now, it's not perfect. There are clearly a lot of places the species is absent when you might have expected to be present based upon those two abiotic niche dimensions. Well, there are other abiotic dimensions in the environment, maybe variability in temperature. There are predators, there are competitors, a lot of things that weren't left out, but there's clearly, information in that empirical quantification of a niche response surface. Now, one interesting thing to me, and this is not something I'll be talking about, but it's the subject of other things I work on. The experiment Richard did was with one clone, okay? Daphnia magna, like all zooplanktons, has a tremendous amount of genetic variability, yet nonetheless, measuring the niche response surface for that one clone had great predictive power about many sites the species is absent from. In other words, across all that genetic variability, there's a kind of conservatism in some basic niche properties. And that's the topic of a whole nother kind of talk I could give on when do you see niche conservatism versus rapid evolution. But that's not. So getting back to COVID, so we've been talking about exponential growth. And one of the shockers to me in this pandemic is how poorly understood the basic concept of exponential growth is, not just by Joe Public, but by supposedly well-educated policymakers in the world, including public health officials. Okay, that was really kind of daunting. Now, when epidemiologists talk about infectious disease dynamics, they don't use little r, which has units of 1 over time. They use r sub naught, the average number of secondary infections caused by an infected individual. And there's subtleties relating these two when there's variation over time and so forth, which we won't talk about. There are also issues in estimation because of things like asymptomatic individuals and so forth. Again, I'm not going to be talking about that. But let's just talk about those measures for a second. I'm going to come right back to this if I have time right at the end of the talk. So here we have our familiar model of exponential growth, where we separate births and deaths, and we don't conflate them into little r immediately. And that's our measure of fitness, right? Instantaneous growth rate has units of 1 over time. Because we have another measure of fitness out there, lifetime reproductive success, which in epidemiology is, the R naught, the number of secondary infections per primary infection, which is B divided by D in a constant environment, which is unitless. It's just it's just a number. How many births do you have before you die? Okay? Lifetime reproductive success in behavioral ecology. And of course, deterministically, in terms of dividing the world into the niche and outside the niche, these two map onto each other, because little r that being positive means big R has to be greater than 1. Little r negative means big R is less than 1. And you can use that to define source habitats and sink habitats with movement and so forth. At the end of this talk, though, I want to suggest that there are interesting subtle implications of differences that these fitness metrics imply for how we conceptualize niches and niche response surfaces. Back to COVID for a few minutes. I mentioned ecological niche models, and people have tried to do that with COVID. So a friend of mine, Miguel Araujo, who's a great macroecologist, biogeographer in Madrid, Spain, put out a preprint in March 12th, 2020, saying that he thought, based upon the data available at the time, doing these, using maxent and those kind of niche modeling approaches, that people in temperate, warm, and cold climates are more vulnerable than those in arid and tropical climates. And immediately there was a response, like 10 days later, from people who I also happen to know on how this is a really bad idea. And I talked to Miguel about this a week or so ago, and he said that he never got around to publishing this preprint because, and he's gotten almost 200 citations, which is more than a lot of actual publications in the literature, right? He says because there was a firestorm of almost abuse on Twitter and Facebook and so forth, social media, accusing him of doing terrible things by putting out ideas like this that could then mislead policymakers. So I was curious, where does that stand today? This isn't the sort of thing I do. So this is a very recent review from a journal where Dr. Natari here looked at temperature dependence in COVID transmission, country by country around the world. And by golly, you use the intrinsic growth rate of COVID as a function of temperature, and there is a thermal signal in it. Now, why is that? In the first place, note that all have little r that's positive, so all are within the niche surface, right? But there's clearly a thermal signal. And so what's going on? Well, what was going on in that debate and then going on at present, the niche of a microparasite like a parasite is defined largely by the abundant spatial dispersion and internal states of its hosts, not so much by external drivers. Doesn't mean they're irrelevant. If you have a vector-borne disease, they're highly relevant. That's what determines mosquito abundance and activity. But of course, even for viruses and bacteria, how long they last as infective propagules could be influenced by the environment, could be influenced by temperature and humidity, by UV, how much sunlight you got, and so forth. But also indirectly, because human behavior can be influenced by the environment. So there could be an indirect causal pathway related to the environment to disease transmission. And this is likely to vary all these factors determining the growth rate of the virus when it's rare, which remember is how we define the niche surface, in a complex way. So I've got a movie here that I hope Nick has come up with, where yellow here, so this go back to mid-March, when people are starting to get nervous before campuses closed down in the US, but it was starting to show up. Yellow is less than 6 cases per million, and the darker it is, the more cases are per million on a log scale. So let's see if we can get this video to work. And this shows the spatial temporal dynamics of this disease, looking at cases per million across the US from that time to the present. And what you can see is there's tremendous spatial heterogeneity and temporal variability, and there's a lot of waves of the infection moving across space. It's very, very obvious when you look at this video, okay? So I want to go on to the next slide now. And now I'm gonna go back quickly to that things I said in that 2009 PNAS paper, revisiting, bringing the Hutchinsonian niche into the 20th century. And I said at the time that there are elements that Hutchinson left out that are really relevant that we need to build into our niche concepts. One is dispersal. Another is stochasticity of 2 flavors, demographic stochasticity and environmental stochasticity. And what I'll be talking about for the next 10 minutes or so, 10, 15 minutes talks, is the interplay of dispersal and temporal variability. But then you have something that, that Giza emphasizes and Liz emphasized a lot in their book, that we're talking about, how species fit together in communities. You have to talk about how they impact the world and how that feeds back upon them. Those feedback loops are absolutely critical. That turns out to be relevant to even Hutchinson's basic ideas of persistence. And I'll say something, just a word about that at the end. And of course, evolution needs to be thrown into the mix, and I'm really not going to say anything about that. So it's very important, and I'm doing a lot of work on it. So, dispersal, and so now I'm going to step away from COVID for a few minutes. I'm gonna go back to these old papers that I mentioned. They're not old to me, but they're old to a lot of you because they were 15 years and so ago. A lot of species live in a patchy environment, and certainly pathogens live in a patchy environment defined by host individuals and populations of those hosts. And if you think about defining, going back to Daphne and Magna, we were looking lake by lake, but we could be talking about the niche at the scale of the core scale of Yorkshire as a whole. Or we could be looking within lake. Daphne is, I'll go back a second, is tiny, a lake, a small lake is millions of times larger than an individual Daphne. So from the point of view of individual Daphne, the lake is actually a very large and complex landscape made up of a lot of pieces that are connected by dispersal that vary for all kinds of reasons, having to do with hydrological flows, lake chemistry, depth in the lake, and so forth. And we can think about defining the niche in this Hutchinsonian sense, the ability to increase when rare, as a function of the environment, but at different spatial scales. And so how do we do that? Well, when you start thinking about that, you have to pay attention to spatial variation in the environment, which is almost always present in the natural world, and to movement. Okay, and so here's a little exercise. And Peter Cheston's talked about things like this in coexistence theory, by the way. And Nick was Peter's graduate student, so we've been thinking about this a lot in the lab. So here we have that lake I've subdivided into grid cells, and there's movement among the grid cells. Each grid cell K has local births and deaths, but there's movement from outside, spillover, and then output to the outside, right? But now we want to characterize the niche of the lake as a whole, kind of averaging over everything. So we just add up all these terms over all the Ks, and we get a change of the average density in the lake. And you can just do some simple, 2 lines of standard statistical terminology and rewrite this as 2 terms. Little r hat is the spatial average over those grid cells of intrinsic growth rates, plus another term that's a covariance term that describes how the spatial heterogeneity and abundance maps onto that spatial heterogeneity and growth rates, right? This is an instantaneous thing, because of course that could be changing over time, right? And all this, of course, could vary with lake chemistry and the like. And so the interesting thing is that this average growth rate over space can be negative, but the overall growth rate over space can be positive if the covariance term is positive. Okay, now, what can we do with that? Well, let's say that the world's temporally heterogeneous. And I'm going to go to temporal heterogeneity and environmental stochasticity a little bit here. Let's say it's temporally fixed, because no matter species when it's rare, there's spatial heterogeneity, no temporal heterogeneity. Well, we have a sort of preferred measure of this V, and that's because if B and D are constant, and then we have density independent movement, this is just a set of linear differential equations. And that matrix will have a right eigenvector, which is the stable patch distribution, comparable to stable H distribution demography, which gives the relative local density found in each patch. And so if you end up, because of the movement pattern, with more individuals found in habitats where the growth rates are higher, and that can happen both with passive dispersal and active dispersal, you've got a positive covariance then between local abundance and local growth. Now, this doesn't have to be the case if dispersal is maladaptive, and I'll show you an example. So here's a little simple theoretical example, and we're going to be talking about a 2-patch model for the pandemic and a little later. So we have exponential growth in each patch, and then just constant density independent dispersal between them. One patch is a sink habitat, the other is a source, and if you had rapid movement, the average growth rate would be minus 0.5, and the population would go to extinction. That's shown on the bottom here. But as you lower the movement, relatively more individuals are found in the high quality patch. That's what this V is. So what have I got? With lower movement, I've got a positive covariance between local growth and relative abundance that allows the overall growth rate to be positive, even though that's this asymptotic growth rate here, even though the spatial average is negative, okay? Now, it doesn't have to be that way. You can have negative covariances. And here's a real world example. One of my favorites, Paul Ketty, a really great plant ecologist, did this work on sea rocket on sand dunes in Nova Scotia, where near the edge of the dune, the sea rockets are sparse. There are a lot of them in the center of the dune. Okay, let's just focus on those 2 subhabitats. Well, he went out and actually measured demography, birth rates and death rates, as a function of density near the sea and in the middle of the dune. In the middle of the dune, death rates are greater than birth rates in a density independent manner across all densities. Why isn't it going extinct? Well, it's because wind is constantly coming off the open ocean, stripping seeds near the margin, depositing them into the center where there's a negative R, but it's not that negative. So you can sustain a substantial sink population there. And now what do you got? You got a low population being depressed by forced immigration by the wind. And you've got a negative covariance then between local growth and the relative fraction of the population found in each subhabitat. If you increase the wind, the population would go extinct. So now we take this and we add temporal variability to the system. This is where I'm going to go back to these old papers. I'm going to try to blast through this really quickly because it's all available. So we're going to start with the really simple scenario where we have a source sink system with unidirectional asymmetric dispersal from the source to the sink, and the source is just fixed. It's just constantly supplying a rain of immigrants into the sink. And then we're going to have exponential growth or decline. And we can talk about density dependence a little later, very briefly. But that might be varying over time. But if it's constant, then the sink settles down to a standing crop of the input rate divided by the absolute value of the rate of decline. So like in Ketty's sea rocket example, there's a lot of input from the outside and there's a modest rate of decline. You can maintain a substantial sink population, okay? Now you make this variable, and we're going to do this. You can do it deterministically or with time averaging or stochastically, and it comes out the same. And so, but you can see pictorially kind of what's going on. So here we have little r, here's 0, way over to the left, it's a really bad sink. And now we got some temporal variability. It's always bad though. But here we have one where it's not too bad a sink, like the sea rocket in the middle of the dune, right? Now you imagine that sometimes there's more rain, so maybe the birth rate is a little higher than the death rate for the sea rocket, but sometimes it's really dry. So you have temporal variability around it. Well, what you get now is you get a tail of positive growth rates, right? And so then in this PNAS paper I had with Andy Gonzalez back in 2000, just a few lines of algebra, you do some time averaging, and you say if there's time variation in that intrinsic growth rate, if you look at what define, the harmonic mean over the time series equals the input divided by the time averaged, arithmetically averaged instantaneous rate of decline. That's the same as you got in a constant environment. Well, what do we know? We know the harmonic mean is always less than the arithmetic mean. So any kind of temporal variation in the sink environment increases the abundance of the sink environment averaged over time. That's what we call an inflationary effect. Now, how big is this effect? That's a qualitative statement. It turns out that depends upon the structure of the temporal variation. So we could compare white noise to red noise, right? Where you have a short-term autocorrelation, so that there's a decay in autocorrelation with time. And what you do when you do that in models, so here we set it up, so in a constant environment, you've got unit input and an average rate of decline of -1. So you have an average abundance density of 1. And then we have variation. And what happens, of course, is we have fluctuations, but the population doesn't go extinct because there's always immigration coming in, maintaining a floor. But when you have good years, you can grow. And when you have runs of good years, which is what you get with a positive correlation, because of the power of exponential growth, you can spike. Notice this is a log scale. So those are time series, and this is just sort of looking at that model kind of average of density on a log scale. It increases with autocorrelation, particularly when you have a lot of variation. Okay, now, so the conclusion is from this model is that temporal variability in the sink increases average abundance there. Ecologists don't believe theory unless somebody's got some kind of data that verifies it. So an experimental demonstration would be real useful. And I happen to be, when I was thinking about this, a visiting professor at Imperial College at Silwood Park, and they stuck me in an office with a bunch of graduate students, which is really great because I was telling them about my ideas. And Andy, who's now a distinguished professor at McGill, he was a grad student with John Lawton at the time. He says, Bob, I'm doing these experiments with paramecium where we're looking at the effect of the color spectrum of temporal variability. He was doing this with Joel Cohen and others on population dynamics. He said, I could just modify this and make it a source sink system. So here's his model organism, paramecium. Here's intrinsic growth. This is a niche response surface. Intrinsic growth rate is a function of temperature. And we'll be dealing with temperatures to the left of about 5 and 10. So there'll be places that'll be times is negative and times is positive. And this is a theoretician's view of his experimental setup. You've got 2 jars and you move them from a source to a sink. But then we're going to be varying the sink. The source is staying constant, okay? First place, can you make a sink? Yes. Well, you keep them at low temperature. There's temperature on the right axis here. Yeah, so like it's below 5. So it's just below 0 growth rate. Here's a sink. This is arithmetic scale on the left, log scale on the right, okay? Now you introduce temporal variability, and we do this by, you throw a random number generator out. Every day you change the temperature. That's white noise, okay? There's no correlation from one day to the next. So it's cold, warm, warm, cold, warm, cold, cold, warm, cold, 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 warm, and so forth. And so then this is the time series you get with immigration. Remember, you've increased the average growth rate a little bit, so you can't really compare it to the constant environment. But what do you see? You see it's now, you've increased the abundance some. That was one of the predictions, that any kind of variability would increase the abundance of the sink. You know, you're up around 100 and you're getting these short periods of exponential growth and decline. On a log scale, remember, that ought to be a straight line. And then, and this was very clever, and I think Andy gets the credit for this idea, you take that same time series and notice you don't have any signs of strong density dependence here, okay? The densities never got high enough that resources ran out. Then you just rearrange the time series, okay? And so what have you got? You've got a time series where the average growth rate's the same, the mean growth rate's the same, but the autocorrelation structure is different. And what do you get? You get runs here. So we got now longer periods where it's good, and you get these exponential spikes, and you can see on the log scale here, it's very nice, and then they decline. But notice you get up in the thousands now. So what do you get? You get what we predicted, which was that temporal variability, particularly if it's positive autocorrelated, can greatly boost the abundance of a sink population. So this is, 20 plus years ago. So now let's go back to the basic scenario. We've got a source feeding a sink. That's what we were talking about. You can imagine this, how is this relevant? Well, if you have a whole bunch of sinks and they're spatially uncorrelated, then a time average equals the space average across them. So you could have a large aggregate sink population maintained by a source. That was fine. But what if all the patches are sink? What if our view of the world is an illusion? You got these immigrants coming in, but they're all being drawn from dispersers, leaving patches that on average is like the one that you're looking at. Can the inflationary effect promote persistence of a metapopulation comprised entirely of sinks? It was not clear, because the only way that would work would be that if individuals leave the patch that they're in, but that lowers that patch's ability to respond with a spike of exponential growth during good times, because it's sending out immigrants elsewhere. It's bleeding your capital. There you are, you got a bank with a high interest rate. You're spending your money, you're never going to have a high bank account, okay? So it wasn't clear that it would work, but the short answer is it does work. And so here we did this in discrete time. This is all published. So there you have the same thing. We can do the spatial averaging again, and again you get this expression for regional growth rate across the metapopulation, which is an average over spatial. They're not technically rates, because it's per generation, plus a covariance term, okay? And it can exceed 1 if the covariance term is there. So here's an example of what it looks like, where your average little r, so that your finite growth rate is e to this, is minus 0.5. So you have a decline of about 30% per generation in a constant environment. And then we have global dispersal with a modest dispersal rate in this case. And we have variance in growth rates on the x-axis and autocorrelation on the y-axis. And what you see is to the right, you get persistence, and to the left, you get extinction. And you could think of these as kind of niche axes in a way, where the niche axes have to do with the statistical structure of the world, not just its mean, but its variability and its autocorrelation structure. And Richard Levins, in a great paper, MAMNAT 1979, he used sort of time averaging ideas to talk about temporal variability and species coexistence. He talked about species eating variance. So this is a species in this system that eats variance in the world. How does it depend upon the rate of dispersal itself? And so this is the global dispersal model, where you go from 0 dispersal to a lot of dispersal.
If nobody, and this is where we have an average rate of decline, arithmetic average of 0.6, that's almost 50% decline per generation. So in a constant environment, you're gone in about three or four generations. If nobody disperses, everybody goes extinct, because that's what every patch experiences. If everybody disperses and you totally mix up, you experience this spatial average. But in between, where you have some dispersal, but not too much, particularly if you have a strong autocorrelation, you can sustain a substantial population size, okay? And again, ecologists aren't gonna believe this unless there's empirical demonstration. So Andy went back to the lab and he made 2 sinks with paramecium, and then he imposed different patterns of temporal variability. And on the top left here, we have 2 patches exchanging immigrants, but they're synchronized in their variation. And this is red noise with dispersal, white noise with dispersal. They both go extinct, okay? These 2 patch metapopulations, if you will. On the bottom right, now you make the patches asynchronous in their temporal variability, and they persist. And then on the left, you make the variability red noise rather than white noise. And notice that you've increased the time average abundance. I think you can see that just eyeballing these graphs. So the effect, you know, you've got a basic metapopulation effect that's augmented by this temporal autocorrelation issue. So what do we got? We've got, if a bunch of sinks coupled by dispersal can persist, if local growth rates are variable, given that you have some periods of positive growth, it requires asynchrony. It's fostered by autocorrelation. I call this stitching the niche, you know, like pulling pieces of space together to make the niche of the species. And what about COVID? And for a long time when I was writing about this, I would write corvid, because I'm a birding. Corvids is a word for jays and crows and magpies. And I got in real trouble the first letter I wrote about it. Okay, so here's in the US a familiar face, Fauci. Says we're a large country that has outbreaks in different regions, different states, different cities. They have different dynamics and different phases. That sounds like what I was just talking about in those theoretical models, doesn't it? So here's a metapopulation picture of the US. These are the urban localities, okay? There's the metapopulation. There's movement among them. There's temporal variability. Here's a snapshot in time. It does, my screen's cut off. I can't quite see with this in, but there's one snapshot. You got some states with big R greater than 1, others that are big R is about equal to 1, others where it's less than 1. Remember, a metapopulation of sinks can persist if there's temporal variability in growth rates. This is showing variability early in the epidemic at the country scale. And of course, there was huge variability in growth rates. And this is something that just came out a few weeks ago. If the variability is asynchronous, and that's just like what I showed you, things are happening in different ways in different places for all kinds of reasons, many of which are behavioral and political in nature. If there's dispersal, there was a lot of movement going on around the US by road, by train, and by plane, even in the midst of the pandemic. This is October, you know, things were getting really bad then. There was still a lot of plane traffic here in the US. And it's magnified by a positive autocorrelation. So here's an estimate in Queens, New York of little r. And of course, over short time scales, at least, there is a positive autocorrelation. So all the ingredients of this inflationary effect appear to be present in the pandemic. So what we did, Nick and Maggie and Greg and Mike and Bert and I, they get the lion's share of the credit, was they actually explored this using epidemiological models with parameters that we tried to make reasonably realistic as best we could, with all the normal caveats you have about how you estimate epidemiological parameters. And so again, we're going to go back to a 2-patch model, you know, 2 localities, Florida and New York, or Tampa and Miami, it doesn't matter, but 2 localities with their own local dynamics and movement between them. And so here and this is like a typical epidemiological model where we have a fixed population to start with. There aren't any births coming in. So individuals just die, the pandemic's going to sweep through. So it's all kind of within a couple of years of a human generation, say. So here's a standard epidemiological model, frequency dependent model, limited number of contacts, it's not density dependence, beta, which will allow to vary with time, movement, then you have loss of infected individuals, and that loss is of 2 sorts. Some individuals sadly die, but some individuals who are infected aren't symptomatic, and so they don't die, but they might move along with, to other patches, and then some recover. Okay, or maybe they're quarantined. It doesn't really matter because this component doesn't have any. So it's a susceptible infected recovered model. And when the infection is rare, what we have is, a little bit, this part, these, when it's rare, then I is small, S is about N. It's basically beta minus these terms. And we just have a model of exponential growth and movement between two patches, just like that example, that simple example I showed you earlier. Okay, but now we're going to let R vary temporally. Okay, not just and why can little r vary in a model like this? Because the variation is social distancing and quarantining. Different polities have different rules about what people do. New York closes down, Florida stays open, for instance. And then we're going to assume very simple temporal pattern and variation. When you can make all these things much more complicated. Effective control is where your local time average is less than 0, and the fluctuating little r, that the average little r is negative. Okay, so if a locality is left to itself, like New Zealand, the virus ought to go extinct. Ineffective control is, of course, where the local time average is positive. So let's look first at square wave variation. So this is something we didn't put in the paper because we had stringent space restrictions. So here we have little r, business as usual. There's an outbreak. Everybody gets nervous. The mayor puts out a dictum saying everybody has to stay home. You have social distancing. And so now case rates go down. And then things will relax. And people say, hey, we don't have any cases out there, right? We set it up so that the average little r is negative. And then we have two patches. Remember, we're going to have this time fluctuating exponential growth or decline of the infectious class in each patch. But the two municipalities or states or countries could be coordinated. It could be trying to cooperate. They're doing the same things. They're mimicking each other, or they're ignoring each other. Florida stays open while New York stays closed, okay? What happens then? Well, let's look at one run where you have one patch where you have an outbreak and the other one you have lockdown. But remember, you have movement. Well, remember, this is exponential growth. And as long as what we've got here is a patch, and we've got modest movement between the patches, the outbreak patch is growing, okay? And because it's feeding migrants to the other patch, it too will start growing along, even though the local growth rate is negative, right? And so now this is what we actually had in the PNAS paper. Now you look at sinusoidal variation. On the left here, we have synchronous control going from a positive R to a negative R in two patches, right? And you can barely see them. There's a green line and a dashed blue line, or a dashed green line and a blue line, I guess it is here. And here's the little r, it's these quantities in the epidemiological model. And now you have effective local control. The average little r is negative, and they're synchronized. And what you get, because they're synchronized, everything's happening in the same way. It's just like Andy Gonzalez's experiment, right? It goes extinct. Okay, you do have outbreaks, but they wane with time. But now you make it asynchronous. So on average, these patches are all the same. Okay, these cities are doing the same thing. They all have the same behavior. They're just not coordinated. And what you get is this pattern where one patch grows and it starts feeding the other one, and then it can take over when it in turn changes, and it can ratchet itself up. This is an example of a larger phenomenon that's very fascinating in science with nonlinear systems like this, called Pandora's paradox. Pandora's, is that right? Parando's paradox. Ellen Hastings wrote a paper about this, where you can have two processes that left on their own are going to the right, but you couple them and they move to the left. Okay, that's kind of what's going on here. You have examples like that in engineering, chemistry, lots of things. But something I didn't realize until we did this, was what happens if you have ineffective control. That means that this doesn't matter for the ultimate. So ineffective control, remember, is where your average R is positive. So now here you have synchrony. Everybody's cooperating, but still you're doomed because it's still sweeping over the landscape. But now you have asynchrony. Neither of you doing it very well. But you can tell now the peak here is, and this is a log scale, is higher. So the epidemic spreads faster because of the asynchrony in control. It's not just persistence, it's how fast you spread, okay? To me that, and so this goes back to the square wave. So to say, how general is this? So this is now a kind of a weak effective local control. This is movement rates from one patch to the other. And this is this social, how much overlap you have. And what you see is it doesn't take very much movement, and you can have disease persistence even with a fair bit of synchrony, not complete synchrony, but a fair bit of synchrony in control measures. So that just sort of restates it again. So merely shifting the timing of social control, things like masking, social distancing, and so forth, can allow disease to persist when otherwise would be eradicated. This shows the magnitude of the effect. Remember, it's sweeping through this population, no new births. So eventually you get herd immunity and the disease disappears, right? But how many people got sick before it's over? And so as you go up here from dark to light, you're getting more synchrony. Was this right? More asynchrony. I said it backwards. More asynchrony. And from left to right, more movement. And you can see both for ineffective local control and effective local control, you have a lot more cumulative cases because of the lack of coordination in this system. Intuitively, why does this effect work? Now I'm just quoting from our paper. Immigration infectious individuals into a location during periods when R is less than 1, a single location during lockdowns, maintains prevalence greater than expected. Those locations can surge to greater prevalence when they switch to R greater than 1. Remember, they'll have R greater than 1 for a while, because that'll be a source then. If different localities enter and leave lockdown at different times, they in effect take turns sourcing and receiving immigrants, fueling the disease spread in each other. So if you go back to the niche idea, the niche of COVID has to be gauged over large spatial scales that include spatial temporal variation, local dynamics, and the pattern of movement among systems. Okay, so we've done some more with things with this that this is something we're continuing to work on. So you can add, make it stochastic. Is it sensitive to adding stochasticity? No. Going from synchronous with a deterministic skeleton to asynchrony, it works very nicely. That's what the bottom ones show. You can make the model more complex epidemiologic. You could have an exposed class that's not yet infectious. And again, if you have asynchrony with movement, it can last for a while and damage a lot of people before it's over. So just at the same time we were submitting this paper, parallel thinking is rife in science. And Nick here, Nick here was one of our students. He got his degree in public health, and then he went with Andy Tatum to Bristol in England, and now he's a professor at Virginia Tech. They published this paper in Science, where they looked at the issue of coordination and exit strategies. When do you stop lockdowns and so forth across Europe? And they concluded that, if you could synchronize intermittent lockdowns across Europe, and half as many lockdown periods would be required to end community transmission continent-wide. Their data was based on, it was a very complicated detailed simulation where they had movement data that they extracted from Google. And this is the magnitude of the effects. So the red line here is these NPIs as non-pharmaceutical interventions, things like masking, lockdowns, and so forth, which are instituted in waves, okay? The red lines is asynchronous. The blue lines are if you synchronize them. So you do everything the same, but you just coordinate your strategies. And you see that as the pandemic goes on, it dies out a lot faster if you had synchrony. And this is sort of like, a running average of new infected individuals. The cumulative case numbers are much larger with asynchrony than with synchrony. So how do we deal with this? In a pandemic, you slow movement. That's what New Zealand did. That's what various places around the world did, Taiwan and so forth. You of course have to control local transmission. You try to reduce these swings and you coordinate. And what did governments actually do? These are from the Washington Post and the BBC. New York had a block by block strategy. I'm sure that sounds brilliant, doesn't it? What did England do? They had a local whack-a-mole strategy. You all know what a mole is. It comes up from the ground. It's like it's a insectivore, and then you whack it, and it goes back underground. At any rate, it didn't work in England. And so there are real practical questions here. So what are we doing now? So we're going to talk about the future a little bit, and then I just want to end with some more conceptual points. So we're working with, we're exploring ideas with Nick, who I mentioned, who's on the ground working with very detailed epidemiological data. Another great epidemiologist we have on staff, Derek Cummins, in my department. And of course, there are lots of questions you can ask about asymmetries, but there are many more populations than just 2 in the world. They're connected in complicated movement networks. Those movement networks themselves vary over time for behavioral reasons as well as for policy reasons. There's asymmetries in population size, there are asymmetries in rates of movement, so on and so forth. And then there's a lot of heterogeneity in hosts. There are asymptomatic cases, there's old versus young, so on and so forth. And so really getting into the epidemiological role requires you to deal with some of these, but we think there's a really exciting possibility. And one of the things we're going to do actually, that I think it'd be very interesting, COVID's not the only disease in the world. There are a lot of infectious diseases translated around, but they're all going to be experiencing the same mobility patterns, but they'll have different epidemiological properties, different transmission functions that describe transmission dynamics. How do their dynamics compare with each other? You know, and maybe they even influence each other in some ways. If you have, you know, heterologous immunity, things like that. And then just the general ecological question, going back to the spatial scaling of how we think of a niche. We need to think about temporal variation in both local conditions and dispersal, and what are the consequences of that for persistence at some spatial scale versus extinction. And so, and then the final thing, of course, you can ask, a lot of the data I was showing you was before these Delta variants and so forth showed up. What are the consequences of these spatial temporal dynamics for evolutionary processes in the pathogen? I think that's a really interesting, important question. When can you get coexistence of alternative strains and that sort of thing? So that's another direction we're going. So I asked our colleague, Nick Ruktananchai up at Virginia Tech about this, and there's some real hints in the phylogenetic data they're getting from molecular techniques that the mobility patterns are really important in what you're seeing. And so I just have a quote I drew from a recent email. And so I want to get back to just one last thing here. And I've talked way too long because it's supposed to be more time for discussion than for speakers to speak. So I've been talking about dispersal and environmental stochasticity, but I want to say a word about demographic stochasticity, because that matters a lot in pandemics and more broadly in the world. In pandemics, it's because when you get everybody who's practically could be infected has gotten infected and recovered, the disease is going to die out in that generation because it's run out of anything to eat. And that's basically demographic stochasticity. You get down to small numbers, fade out. So I'm going to say something about that. Density dependence matters because, having a high R, high growth rate, doesn't mean you're likely to persist if you're a pathogen, because you can really rapidly burn through a local population and eat out the susceptible host and crash to extinction, sort of like the reindeer deer of St. Matthew's Island. So let me, I'm going backwards. Let me go forward. So we're gonna go back to this little, we got these 2 fitness metrics. And now we want to talk about and go back to more general issues about what the ecological niche is. And remember, I think it's useful to think about the whole niche response surface, not just the border here. And here's a little diagram that I find useful. And this just says birth rates and death rates, okay? And let's look at big R and little r for those. Well, when a population is at equilibrium, that's the line of slope 1, little r equals 0, births equal deaths, or r not equal to 1. But if you're away from that population that can grow potentially or decline, if you look at the family of parallel lines, that's all possible values for the intrinsic growth rate, right? D minus D, because the distance between here and here is a constant because the lines are parallel. But what's the family of lines to the origin? Well, that's R naught, where the slope of the line is 1 over D. So for any non-equilibrial little r, there's an infinite number of values of R naught that go along with it, and vice versa. Does that matter? Okay. Well, so here let's talk about what the niche surface might look like a little bit. Well, here we have, remember, you go back in births and deaths rather than going to little r immediately. And here we have 2 abstract environmental axes, X and Y, and we have a Gaussian bulging out of us with an optimal birth rate here at the cross. And here we have death rate as a function of the environment, but the optimal environment for deaths is different from the optimal environments for births. So now we look at the niche response surfaces you get from little r versus big R. And the borders are the same. That's the yellow here, but the optima are different. okay? And the shape of the niche response surface is quite different. The marginal effect of an increase in your fitness measure with a given change in where you are in the environment. Now, does this matter? I mean, maybe that's just, so what? Well, it does matter if transient dynamics are important and numbers get low, and the world is always in transition. This is what the world looked like 25,000 years ago. Giza, you're here in Hungary, and Liz is way over here someplace. You're on the edge of not far from permanent ice and tundra. Every species that's around you today in Hungary wasn't there 25 years ago. And when they came in, they were probably rare. When species are rare, demographic stochasticity looms large. And we know from basic extinction biology science, going back to Kendall, 1948, he had this beautiful paper on birth-death processes in continuous and fluctuating environment. But we're gonna let the environment be constant for a second. And you work out the probability of survival of a clone introduced into new habitat. And if the birth rate is less than the death rate, the probability of survival is 0. If the birth rate is greater than the death rate, it's determined by big R, okay? Little r matters in a transient period, if you haven't let t go to infinity, okay? And then if carrying capacities are low, demographic stochasticity continues to loom large. That was for exponential growth. That's an idea that goes back to McCarthy and Wilson before, because if you're low carrying capacity, you can random walk to extinction. So this is probability of persistence for an asexual colonist within its niche. And you can see if its little r is low and its death rate is high, it's going to have a big R very near 1, so it has a very low probability of persistence. And this is what its niche response surface would look like if you'd expressed it in terms of probability of persistence. And that differs quite substantially from the niche response surface you would expect based upon little r. I'll skip that in the interest of time. And now none of that might matter if dispersal is totally unlimited, because eventually if the probability of making it is non-zero, you'll have somebody hitting it luckily. But the bottom line, and this is all, I think my last talk before my concluding slide, the bottom line in Hutchinson 1958, the niche is environments where persistent indefinite. That is not the same as the niche is the environment when you think about the intrinsic growth rates, okay? Because to have long-term persistence, you have to pay attention to stochasticity, demographic stochasticity, environmental stochasticity. You have to pay attention to feedbacks, because if feedbacks are too strong, like in that case of a pathogen with a big R, it gets in and it just eats out its host supply, and then it wipes itself out. All those are melded into having probability of persistence. And if you go back to things like ecological niche models, of which there are, I think something like 8,000 to 10,000 papers in the literature using those now, that are using presence-absence data and estimating the niche, what they're actually looking at is something closer to the probability of persistence than to intrinsic growth rate per se. Intrinsic growth rate is still really absolutely critical, important. But there's a distinction between these two niche concepts that I think would be worthwhile people paying more attention to. With that, that's my last slide, and I've talked way too long, but I sort of had to get through these ideas. And I hope you don't mind my having rambled on for, gosh, an hour. My goodness. Thank you very much for your attention. And I will stop sharing. I took that photo, by the way, of a curious lemur in Madagascar a few years ago. I'm so yearning for the day we can travel again. So,
thank you so much. Do you have time to take any questions?
Of course, I've got time to take questions.
So if anyone has any questions, in the reactions box at the bottom, click raise hand, and then we'll sort it out and put them forward.
Should I read them, or?
No, they'll come. Should we start
They're complimenting me, but I won't read those aloud.
Matthew Spencer, do you want to unmute and ask your question?
Yeah, so I really enjoyed that. Thank you.
Thank you, Matthew. Good to see you.
The question I was going to ask, you kind of somewhat answered right at the end. So right at the start, you said that Hutchinson's original niche concept was set theoretic, and yet, that's kind of maybe page 1 of a book on set theory. And I was thinking that pages 2 through to 200 don't seem to really tell you anything that you need to know to be an ecologist. But then right at the end, you came back to this thing about niche response surfaces based on little r and big R being quite different. So can you get any new theory by thinking about the kinds of operations that you perform on sets by the continuous map between those two things?
Well, for first place, I mean, historically, I think saying that yes, he had a kind of set theoretic view of things in 1958 is accurate, because he described, these, his hypervolumes and so forth. But thinking about the probability of persistence, to me, the question is, what are the ingredients that go into probability of persistence as a function of environments? What all do you have to pay attention to? Okay. And one of the things you have to pay attention to, first place, if you think about big R, B divided by D, I mean, think of it very simply. So if you have big R, if you have a closed population and big R is less than 1, then you know that extinction is inevitable. But it's not guaranteed if big R is greater than 1. So one thing you'll want to do is to say, if you're trying to relate demography to species distributions, you'd like to keep track of births and deaths separately as long as you can before you combine them into your fitness metric, because they can be combined in different ways that give you different information about the world. What big R gives you, what little r gives you, is how fast stuff is flowing into the population from the environment. Okay, so if you're doing a mass balance kind of thing, that would tell you how much stuff is going into this growing population, right? That's really valuable. What big R do is pure replication. It's how many births you give before you give death, okay? And so think about a little r equal to 2, right? Well, I can construct that with B's and D's that are different. So let's say I have a B of 102 and a D of 100. Remember, those are instantaneous continuous time measurements. Okay, so B minus D is equal to 2. Will you go extinct according to Kendall and branching process theories? It's very likely you'll go extinct, because little big R is nearly 1. You're very likely to random walk to extinct if you just throw a few individuals in. What if you have a B of 2 and a D of 0? You still have a little r of 2. Extinction will never happen because nobody dies. So there's a cartoon version of how you can have massive differences in likelihood of extinction for clones that all share the same little r. And then I would argue that if you're interested, like many of us are, in geographical ecology and where species persist and their ability to invade and increase when rare, that's really important. So that was one point. Let's see, there were others I felt. Oh, the other thing is that probability of persistence includes more than your ability to increase when rare, because you have to say, well, how many are there to start with? Because the probability of persistence may, if you have a B of 102 and a D of 100, little r of 2, you're likely to go extinct if you start out with 1 to 5 individuals, but you start out with 5 million individuals, you'll never go extinct. So probability of persistence depends upon some kind of specification of initial conditions. It depends upon specification of the time scale, because in the long run, in a bounded world, extinction's inevitable. So you have two things that you need to specify if you're talking about probability of persistence: initial conditions, and what's the time scale over which you're thinking about it, right? And that's often not explicit when people talk about these things. And you have to say, okay, I mean, in Kendall's case, he was talking about, are you talking about timescales long enough where you have to talk about environmental stochasticity as well as demographic stochasticity, so on and so forth. And then you have to start talking about the feedback dimension of the niche influences the probability of persistence, because that's what determines carrying capacity. And we know that low carrying capacities, and this goes back to MacArthur and Wilson, go along with an elevated probability of local extinction. Okay, so you get up to that K very quickly, and then you bounce around and you might random walk to extinction. So there's a kind of interdependence of the impact and feedback dimensions in the niche, or whatever you want to call it, and determine the probability of persistence. Okay, and I just think these are points that have been kind of missed, and these are all also actually relevant to competitive interactions among species. And there's been a spate of recent papers, very recent papers saying, hey, look at it, just the ability to increase in rare in kind of a deterministic model doesn't quite capture what you need for long-term coexistence if you take into account demographic and environmental stochasticity. That was a very long answer to your question, Matthew. I'm sorry. But I'm a long-winded kind of guy, so you guys shouldn't be surprised. Oh, I have no idea whose hand up was next.
I think it was Michael Hochberg's next.
Michael.
Hey, Bob.
Hey.
Yes, great talk, very stimulating. So I'm interested in the actual applications to SARS-CoV-2, and in particular, the dependence of your models on migration rates, migration distance. And aside from the invasion of new variants of SARS-CoV-2, what kind of evidence do you have that migration has any impact whatsoever on the spatial dynamics?
So I mentioned this paper that's out in Science by our colleague at Virginia Tech. And he had, I think, a very strong argument there. And it was not based upon, like theoretical ecology. They had models where they were looking at each locality in Europe. And I can't remember, they had some that were by state, not by state, by country, by city and so forth. And they looked at the actual pattern, estimates of local big R in those locations. And then they had actual movement patterns. And so they then asked they did a thought experiment. You look at the actual realized pattern of the COVID, and then what would have happened given those models that they were trying to tailor to the actual movement patterns and leave the movement patterns the same, but then synchronize the movement, synchronize the control measures. And they found that there was a great shortening of the time to control the pandemic in Europe as a whole. So I'd recommend you read that paper, and we're going to be trying to work with that Nick, as well as our Nick, to think about it more concretely. So what we did in our PNAS paper was pointing out an abstract possibility, something that was woefully underappreciated by some of the world's best epidemiologists, that if you have an epidemic in space and things are happening differently in different places, and then you have movement, that the overall dynamics isn't given to you by just simply adding up all the local dynamics. That was the conceptual message of that paper, which I think was really missed by policymakers here in the US in particular. Do I have any evidence that mobility matters? No, not myself. I have a hard time reading, figuring out how to get that kind of evidence from epidemiological case data. But we're trying to work with people who do. Okay.
Right. So I'll read that paper, but I guess my point is that there's no evidence that I've seen that on city scales, the epidemic is burnt out. So there's always existing cases. And the question is, to what extent can movement swamp effectively the local epidemiological dynamics of SARS-CoV-2, aside from the actual invasion of new variants where it could? So I will read the Science paper, but I'm a priori very skeptical that this is having any effect whatsoever.
Yeah, the idea isn't that it swamps local transmission dynamics, but it sort of jumpstarts local transmission dynamics, where you can have an influx of disease carriers, people who are infectious at the time. And then when conditions change, the disease spreads quicker because it has a head start, because they're more infectious individuals than expected. Based on if you
right, sure, but once it's there, it stays. Do you have any evidence of burnout at anything but the most local scales, let's say in a neighborhood? That's possible, but
it doesn't require that the disease.
Remember, we're also making a point where there's the issue of, does it persist or go extinct? And that ties into kind of the niche concept, right? What are the environments that permit persistence? But there was the other effect, which is it's not burning out on average any place, which is sort of like what you're saying, but still movement among localities can amplify the overall spread of the pandemic across the entire metapopulation. That was the lower panel of one of those panels from our PNAS paper, which I hadn't picked up on before, so that the invasion of a species into new habitat can be amplified because of this kind of spatial temporal dynamic. And in your case, the disease isn't disappearing anyplace locally, but its overall extent is still amplified by these sorts of effects. At least that's a possibility that
Okay, I'll look at the papers.
worrying about. Right.
Great.
Yeah, just send me an email, Mike. I'd be happy to send them to you.
Sure.
Okay. Let's see. There were Mike Axel has his hand up. Hi, Axel.
Yeah. Sorry. So thanks for the talk. And you said if people would have, I have two questions actually, one for you and one for everybody. And you said if people would have listened to what you said, then lots of life could have been saved. As I understand this right.
That's just vanity on my part.
Yes, but let's assume you're right. Let's assume you're guys. You really had great ideas and you could have saved a lot of lives. And my first question is, okay, so how many lives have you already saved? And the second question is, if it's not so large and it's to everybody, what are we doing wrong? Why are people who invent a new pill against COVID or a vaccine, why do they get so much attention and people listen to them? And why don't we? And why do people follow our advice?
Do you have any examples of important people in the world listening to theoretical ecologists, Axel?
Yes, but that's the point I'm trying to make here. We're doing something wrong.
Yeah, they should. I said at the beginning, that's why it shocked me how few people clearly understood just basic ideas of exponential growth. Okay, because if you have an exponentially growing process, you don't wait till it's really obvious before you start getting onto it. You got an invasive species, you get out there and you try to pull up all those weeds immediately. You don't wait till it covers half your landscape, right? And that's true for pandemics too. You got to squash them early. And yet people saying, oh, it's not a problem. We've only got a few hundred cases, you know. And so there's a kind of, and of course that shows up in spades. You know, think how, I mean, it's another arena, economics, how many people don't understand interest rates on their bank accounts. And therefore they get screwed with mortgages and so forth because they just don't understand the basic concept of compound interest. So that's part of it. So if you don't understand that, you're not going to get any of these subtleties I'm talking about, right? You know, and what people do identify with are measures that you know, we are better at thinking about the world as individuals than at how populations work. You have to learn how populations work, learn population thinking. We're better at learning a treatment for yourself, a vaccination, a pill you pop, right? Everybody can identify with that. These things at the population level, I think it's very hard. I think it's just hard for people. I think it's a failure of educational system not to teach these concepts. I think they ought to have exponential growth in elementary school myself. I mean, you can teach it with all kinds of board games and stuff. But that's sort of my, you know, I'll get off my
okay. I will keep the question up for everybody else. Is there anything else we could do to make a bigger impact with our work?
Well, I mean, you know, it'd be interesting to know. There are some countries that have been very successful in this pandemic, and it'd be really interesting to know what they did. Taiwan has, you know, a pretty big population, and they've had very, very few cases and deaths relative to either the EU or the US on a per capita basis. And it would be interesting to know if, and their vice president's an epidemiologist, if in their think at all, even implicitly, I mean, anytime you quarantine a locality, you're talking about something related to us, because cutting out movement entirely makes our effects goes away. That's what quarantine is, right? That's kind of an extreme measure. You know, it'd be interesting to know if they, you know, how much of kind of manipulating mobility patterns was involved in their public health responses that allowed them to control it so effectively. But maybe it's irrelevant. Maybe it was all masking and contact tracing and so forth. I mean, those are really great questions, Axel. And if we knew the answer to it, we'd have mega grants and, you know, fly first class to Europe and so forth.
The colleague behind you is raising the hand, by the way.
Somebody else I thought had their hand up, but I've been going on for quite a while. So, yeah, Giza, did you have your hand up earlier and you just gave up?
Thank you, Bob.
Maybe Giza's gone to sleep.
I think it was Jack who had his hand up and he put it down.
Yeah, I can't remember. Somebody else had that.
I don't know, I guess we might have possibly reached the end of the questions, but if there's anyone else who does have anything, now's the chance. So if you don't know, it's the reactions box down the bottom, the little smiley face with the plus. Click that and then click raise hand if you want to. But
Yeah, thank you everybody for listening. I appreciate it.
Oh, looks like nobody. So yeah, well, thank you very much again, Bob. It's been.
And the next
really great.
lecturer is Jeremy Fox.
Yes, we don't know the title yet, I'm afraid. I don't think either. But it should be Jeremy Fox, who used to run the Dynamic Ecology blog, if you've come across him there. Yeah, thank you so much, Bob.
Thank you, Chris. Thank you for running it. Thank you, Axel and Gazer and Chris for the invitation. Appreciate it.
Thank you.
Bye-bye, everybody.